Hello, today we'll talk about the production of erythropoietin by fermentation, also known as old biotechnology, and also its production by new biotechnology. So what do we know about erythropoietin? Erythropoietin, also known as EPO, is a glycoprotein hormone that controls erythropoiesis, or red blood cell production. It is produced by interstitial fibroblasts in the kidney in close association with peritubular capillary and tubular epithelial tubule. It is also produced in the perisinusoidal cells in the liver. Its main role is to induce terminal differentiation of erythroid precursors into red blood cells. These kidney cells are sensitive to low blood oxygen content and will release EPO when oxygen is low. After being released into the bloodstream, it binds with receptors in the bone marrow where it stimulates the production of red blood cells to increase the oxygen carrying. Medically, EPO is used to treat certain forms of anemia, polycythemia, and so on. For your information, the human EPO has a molecular weight of 34 kilodaltons. A predicted structure of glycosylated human erythropoietin is illustrated here. Having said this, we will now take a brief look at the history of erythropoietin. In 1978, Burt and Jordanet proposed that the symptoms of anemia and mountain sickness resulted from hypoxia. In 1906, Carnot and Deflandre observed that serum from an anemic donor rabbit injected into normal rabbits resulted in the increase of erythropoiesis. Years later, Erslev elegantly demonstrated that plasma from anemic rabbits contained a factor capable of stimulating erythropoiesis. A few years later, Goldwasser and colleagues showed in a series of papers that erythropoiesis was governed by the relationship between oxygen supply and demand, and identified the kidney as the site of this regulation. It took 20 years to purify few milligrams of erythropoietin from over 2,500 liters of urine from patients with aplastic anemia. This massive effort provided by the basis for the cloning of the human erythropoietin gene in 1985 and the possibility for large-scale production. Within 2 years of the successful cloning of the gene, the first trial demonstrating that recombinant human EPO stimulated erythropoiesis in humans was established, and in 1989 the US Food and Drug Administration approved its use in chronic renal failure patients receiving dialysis. In 1990, the FDA approved erythropoietin for use in chronic renal failure patients not receiving dialysis and in patients with the HIV and anemia zidovudine. The method for production of human erythropoietin, comprising of culturing human erythropoietin producing mammalian cells in a serum-free medium with repetitive medium exchanges, wherein each of the medium exchanges is carried out by collecting 80 to 95% of the culture. When the density of the viable cells in the culture has reached 2 by 10 raised to power 6 to 4 by 10 raised to power 6 cells per milliliters. The medium exchange and culture are then repeated 4 times. The steps comprises of applying the culture supernatant obtained by the method for production of human erythropoietin to dye affinity column chromatography and collecting a fraction containing human erythropoietin activity. Secondly, applying the collected fraction to hydroxyapatite column chromatography and collecting the fraction containing human erythropoietin. Next, applying the fraction collected into the step above to cation exchange chromatography and collecting a fraction containing human erythropoietin activity. And lastly, applying the fraction collected previously to gel filtration column chromatography and collecting the fraction containing human erythropoietin activity for the last time. Next, we'll talk about the issues that arise from the production of recombinant human erythropoietin. If during the fermentation process the medium is invaded by foreign microbes, there will be a loss of productivity, recovery process will be difficult, and the contaminant may degrade the final product. Also, there may be DNA degrading activities in the fermentation broth and in the wastewater that may lead to rapid destruction of plasmid DNA added to the medium. During the purification process of erythropoietin, there may also be the presence of microbiological contaminants. Other than that, the capacity of the cells for producing the recombinant proteins is not fully utilized in general. Aside from technical process parameters such as nutrient supply by way of the medium composition, the cell line specific properties, which may be expressed as different growth rates, production kinetics, cell vitality, post-translational processing for glycosylation and sialylation, also play a central part in the terms of product quality of the EPO obtained and the overall productivity of the fermentation process. Let's now look at how new biotechnology has helped to rectify some of the issues in erythropoietin production. There have been ongoing improvements in the production of erythropoietin. The human kidney cells produce erythropoietin, which can be isolated from urine. In 1977, milligram amount of EPO was successfully isolated and purified from 1,500 liters of urine from patients with anemia. The use of transgenic organisms instead of cultured cells as bioreactors is a tactic to mass produce erythropoietin more efficiently and thus at a lower cost. To prevent the contamination problem which might occur during fermentation, the following stages are involved. First, we identify the gene that produces erythropoietin, isolation of the gene, replication of the gene in an easily manipulated microorganism, production and purification of recombinant erythropoietin in a stable biological form, large-scale production of recombinant erythropoietin gene into Chinese hamster ovary cells, which were then able to produce recombinant erythropoietin. And finally, the recombinant EPO can then further be produced in a large quantity using fermentation. Now, let's go on to consider some of the implications in the production of recombinant erythropoietin, also known as REPO. Recombinant erythropoietin has been used for 20 years in anemic patients undergoing renal dialysis, and it is now approved for the use of autologous blood donation and to reduce transfusion requirements in patients undergoing major surgery. People are concerned about animal welfare and biotechnology's redefinition of the relationship between humans and animals. Drugs, in this case EPO, purified from animal milk or blood requires exceptional levels of safety testing before animal and human health concerns are addressed in the satisfaction of the consumers. There is an important and urgent need to consider clarify, and where necessary, strengthen the ethical and legal framework within the clinical and research uses of human tissue. The ethical issues related directly to the core of respect for human beings, namely that they and their bodies should not be injured and that nothing should be done to them and their bodies without their consent. Besides the process involved in production of recombinant erythropoietin takes a long period of time and gives a low success rate. Thus, developing transgenic animals is currently very expensive. In cancer therapy, epoetin alpha could activate intracellular signaling pathways directly in tumors that express EPO-R. In addition, the ability of the EPO to stimulate new blood vessel formation could enhance perfusion to tumors and support their growth, or alternatively potentiate anti-tumor therapies by enhancing blood delivery. EPO has its dangers. EPO injections thicken the blood, which increases the strain on the heart. It is dangerous because it will slow down the heart rate while asleep. The increased thickness or viscosity of the blood increases the risk of blood clots, heart attacks, and strokes. Other than that, the ethical dilemma in healthcare that arises from biotechnology is its cost and access to new treatments. A number of studies in several types of cancers have shown that patients taking erythropoietin did not live as long as other patients. Furthermore, same synthetic erythropoietin is also being used by some athletes as a form of blood doping. There is an active black market in EPO, and in addition to the legitimate EPO preparations for medical use, there are counterfeits that may have few, if any, active ingredients and carry the risk of contamination. That's all for now for the presentation of recombinant human erythropoietin. Thank you for watching our presentation.